Casper Mossman. I'm Communications and Marketing Director at QB3, and I'm hosting the webinar today. We have Kelly Harkins Kincaid, CEO and co-founder of Astrea Forensics, as well as Claret Bioscience, former UC Santa Cruz postdoc. So what are we going to hear about? Well, as I've just been sort of ranting about, or we're going to learn about forensic genetics and the extraction of genetic information from evidence, which is much more sort of impoverished than you would think you could possibly getting any useful information out of. So, as I mentioned, I am a fan of this terrible TV show, Cold Case Files. I don't. It might not even be made anymore, but it's this endless series of cases in which investigators go and they sequence some DNA evidence that was in the kit from whatever crime happened 30 or 40 or 50 years ago, and then lo and behold they find out who it is, and then they or they just tell the story. But the thing is that they're working from these kits, and what the viewer, of course, doesn't recognize or can't be really expected to know is there has to be usable DNA in the kits, and in some cases it's there's lots of evidence and you get to see it, whatever. But in again, a lot of cases, especially in cases of unknown identity, and maybe there's not even a crime, but it's an accident, and families are looking for closure, and it turns out that there are techniques to get this information out of evidence, which is, I don't know, we're going to learn about rootless hairs today, but teeth or things that you'd think, well, where's the DNA? And if there is DNA, maybe it's degraded with age or the conditions that it was stored in. So we've invited Kelly Harkins Kincaid, who a former UC Santa Cruz postdoc, and she did PhD work in Arizona State that combined paleopathology and bioarchaeology and genetics to recover and reconstruct the genomes of ancient pathogens that infected once living humans. Worked primarily with TB and leishmaniasis in the Americas, but also experience, I don't even really want to know about this, with the genomes of plague and syphilis. Fun stuff.
Very exciting yes.
Genomes. Yeah. Okay. So currently running two genetics companies. One was not enough, Astrea and Claret Bioscience. She may mention that Claret is looking to hire a salesperson with deep knowledge of next generation sequencing. If you're interested, you should get in touch. This is in the near future. And involved with, as PI and CEO of both these companies, involved with design, validation, and commercialization of these next generation sequencing tools for application in the clinic or law enforcement related use. Kelly, it's great to have you.
Yeah, thanks for inviting me. It's good to see that there's some participants rolling in. So I'll share my screen and just get started. So thanks for the introduction and the invitation, of course. I'll be talking today mostly about the one startup that does work in the forensics kind of realm, which is called Astrea Forensics. What we do is provide genetic services to law enforcement to aid them in the identification of unknown persons. And sometimes that's helping to identify remains, unidentified remains, and sometimes it's for helping to generate leads for their investigations. But a little bit of background. So this company, as well as Claret Bioscience, the other startup that I run, was co-founded by Professor Ed Green at UC Santa Cruz. He's faculty in biomolecular engineering and bioinformatics. And we co-founded Astrea specifically in 2019 to apply ancient DNA techniques to samples from forensics contexts. This was work that had started in his academic lab, where I was a postdoctoral fellow for a few years, starting in 2014. And the forensic work didn't really have a home in the academic lab, and it was sort of becoming overwhelming. So we spun it out into Astrea. It was a natural home for the technology that we were using because Claret Bioscience also uses a similar, if not in some aspects, the same next generation sequencing technology. So both of our expertise and background, or some of our expertise, he's got a lot of expertises, but it is in paleogenomics, or the study of ancient DNA. My training specifically is in archaeological genetics. My PhD is actually in anthropology, whereas Ed's kind of early claim to fame was being known for the guy who sequenced the Neanderthal genome, until very recently, when he started to become well known for something else, which is sequencing the DNA that's contained in hair, mostly from cold cases. So this is a New York Times article a year or two ago. So I'm going to talk a little bit about how we got to this, what, you know, kind of the origin story of Astrea, and I'll talk about genetic genealogy as you know, in general. I'll talk a little bit about how these methods of genetic genealogy and the methods that we use with whole genome sequencing, how they differ from forensic, traditional forensic testing, and then I'll talk a little bit about the specifics of our technology and what we do. So the origin story of Astrea starts in a San Francisco basement in 2016. So there's a couple in San Francisco who were renovating their house, and in the basement, they discovered a coffin. And this coffin was actually pretty unique in that it had a glass front. And inside, you could see that there was a young girl buried. She was very well preserved. This casket was kind of hermetically sealed. Unfortunately, I think that the guys who were working at the house and discovered the casket opened it, and so the remains started to degrade pretty quickly. You can kind of see that there's already mold forming on the child's face in the photo, but she was very well preserved. She had a lot of hair, and this Garden of Innocence project got involved, which is an organization that kind of takes ownership of abandoned or unidentified children. And, you know, of course, the question is, well, who is this little girl who the Garden of Innocence project named Miranda Eve? So a team was assembled of volunteers, historical archaeologists, scientists, genealogists, and they very quickly discovered that the area where this house is currently located was where the Odd Fellows Cemetery used to be. That cemetery was relocated, like all cemeteries in the San Francisco Bay Area, to Colma, and this little girl was left behind. So here's an image from San Francisco in 1935. This is about where that Oddfellow Cemetery was. Here is San Francisco today. And so the team was able to sort of put composite photos together with the original maps of the cemetery and really narrow down where in the cemetery this little girl could have been buried. And they used the kind of estimated age of that glass coffin, which is a very specific coffin that was made only in a certain kind of couple of decades. And they used the cemetery records and death records to narrow it down to a few possible children. And so the genealogists, meanwhile, they're sending hair to the paleogenomics lab to try to get enough DNA information out of the hair to be able to use in a comparison should the genealogist find a living relative, right? So the hair was pretty poorly preserved. What we're seeing here on the left is a plot of deamination. This happens as DNA degrades. The cytosines lose their amine group, and they when the PCR amplification is occurring, the polymerase reads a deaminated cytosine like a uracil. So it actually misincorporates a T where there should have been a C. And so in the sequence data, you can read this as a C to T transition or damage. And this is kind of an indication that the sample is indeed very old and very degraded. The DNA pieces, so the DNA fragments within the hair are also very short. This is sort of our first look into a more detailed fragment analysis of the DNA in hair shafts. The other part about Miranda Eve's hair was that it was only about a little bit less than 10% endogenous. So that means that of all of the DNA sequence reads, 90% of them were not human or not able to be mapped well to the human genome. Either they were too short or they were actually different organisms, mostly fungus and bacteria, which you could see from that previous slide was obviously becoming an issue. However, nonetheless, they were able to generate genome-wide data and had some information about heritage and would eventually have data to use in comparison. So the genealogist narrowed down the list of possible young girls who died around the 1870s. And one of these girls, one of these individuals, is Edith Howard Cook. The genealogists then did their thing and they built out a family tree. And of course, what would be needed to kind of make this match possible is a living relative. And so Peter Cook here, I think, was in his 80s when they found him and contacted him and say, hey, here's the kind of interesting project that we are trying to solve. Will you do one of these direct-to-consumer tests and provide data for us to, actually, no, it wasn't direct-to-consumer tests. I think they just went ahead and did whole genome sequencing on Peter Cook at the time. So that was all done in the paleogenomics lab at UC Santa Cruz. So then, of course, is the question, well, what is the expected relationship? So here we're looking at a great nephew, right? What is the expected relationship or shared ancestry between these two individuals if this was indeed the correct relationship? So these kinds of charts are useful. They're used a lot in genetic genealogy. They're usually in centimorgans and not percent ancestry, but this is still useful for these purposes. It works fine for this illustration, but as a centimorgan, they're usually in, a centimorgan is kind of on average about 1 megabase of DNA. And we'll get into that probably a little later, but so now we kind of have an expectation of what the shared ancestry between Edith Cook and this great grand nephew would be, or grandnephew would be, and it's about 12.5%. So what we have here is our EDS analyses plotting the probability that a segment of DNA has IBD, or identity by descent, as opposed to identity by state. So this is an indication that there's a shared genetic relationship. The scale here is a p-value. The p-value of being IBD, where blue is very likely and red is not likely. So what we have on the top is a positive known grandparent-grandchild relationship, which should be about 25% shared ancestry. On the bottom is two unrelated individuals. So remember we're expecting about 12.5% of a relationship between these two individuals, and that is indeed about what they were able to observe when comparing the hair from Miranda Eve and Peter Cook. So that was the success story that allowed them to identify this little girl. She was reburied, and you can read a lot more about this at the Garden of Innocence website. So once this hit the news, it didn't take long for someone to connect the dots. And that someone was Barbara Ray Venter, the genealogist who's most well known now, of course, for identifying Joseph D'Angelo, the Golden State Killer. And she said that when she read the article about this genealogy and the DNA from hair, she said her eyes lit up, and she called Ed and she basically said, I have some hair for you to try. And what that hair was from the victims of the Barrett Brook murders. If you're into true crime kind of podcasts or, television shows, you might have heard of this. There is a very nice podcast that you can Google and find. Ed had begun, his foray into forensics in his academic lab through this connection with Barbara. So the cases slowly started to trickle into Ed's academic lab until it became kind of overwhelming. And at that point, we spun it out into Astrea. We already had Claret Bioscience, so it was a kind of a nice, easy transition for the technology to come into this startup. And my background being archaeological genetics, it felt also like the proper home for it. So now I'm going to back up a little bit and talk about, more generally about this field that Astrea is working within, which is investigative genetic genealogy or forensic genetic genealogy. IGG is how I'll refer to it. So it really, genetic genealogy, with or without the investigative part, relies on this boom in the direct-to-consumer market, where these companies are offering genetic tests to the public, and there are associated consumer databases containing the output from those genetic tests. Now, in the forensic context of forensics, genetic genealogists then use those profiles that are generated instead from spit samples, instead from crime scene samples or unidentified human remains, to identify close genetic matches. And then these are used by law enforcement to generate investigational leads. The United States Department of Justice has really tried to provide guidelines for the use of IGG. And basically, when all else fails, when you do all of the right methods, the standard acceptable methods, when those all fail, then you can turn to IGG. So that's kind of the framework that we are working within currently. I've underlined these terms because, this is kind of the way that you might see it written in the news or in popular writing. But to me, as someone who's in genetics, when I see they're taking a DNA test, there are DNA profiles. What exactly do you mean by that? What genetic markers are being tested? What format is it in? What technology is being used to assay those markers? What kind of matching algorithms are being used? So I'll get into that a little bit. So there are a handful of direct-to-consumer genetic testing companies. Not all of them are listed here, but these are the main ones. And these are companies who own your DNA data, and they host that data in databases that are controlled by them, owned by them, and have their own algorithms for doing IBD or doing segment matching, to provide you with the cool user interfaces that you're probably at least familiar with, if not, if you haven't done it yourself. The outlier here I want to point out is a database called GEDmatch. GEDmatch, it's not a DNA testing company. It's an open source platform where you can upload your own personal genotype data. So if you take a 23andMe or an Ancestry test, you can download your data and then take it over to GEDmatch and upload it yourself. This allows people from testing at all different platforms to be able to try to find more relatives who might not necessarily be in the database that they tested with. It's really important to note that there are only 2 databases that are available for law enforcement uploads, and those you have to opt in to the law enforcement searches. So that's just a bit of information there. Okay, so to give you a sense of just how different it is, how different the things we do at Astrea are in the context of forensics and DNA testing, I wanted to give you a high-level overview of traditional forensic testing and human identification, as well as what kinds of testing genetic genealogy relies upon. So forensic crime labs, they assay STR markers. These are length polymorphisms. It stands for single tandem repeats. And the DTC companies use genotype arrays to assay SNPs. And they're specific in the subset of SNPs that they assay because they are looking for a curated set that has high alternate allele frequencies, or else if they use the kinds of genetic variation that is most common, which is rare variation, it wouldn't be very useful for connecting people through shared ancestry. What we do at Astrea is we do whole genome sequencing with next generation sequencing platforms, Illumina mostly, exclusively. And we are attempting to build genotype files that are based on the panels of SNPs that the direct-to-consumers testing uses, but all of the direct-to-consumer testing companies use different kinds of panels. And so what we can do is we can call any site in the human genome. And so we can, of course, just choose what SNPs we want to be able to report in our genotype. So the number of markers that are called or are being assayed by SDRs is just a handful. And the panels that the direct-to-consumer tests use are in the hundreds of thousands. And of course, we can call millions of SNPs if we so choose. Now, SDRs are really different than SNPs. They're microsatellites. They're, like I mentioned, length polymorphisms. They're in non-coding regions, and they're chosen for forensic purposes purposefully so that they convey very little information about a person. They don't carry information about phenotype or ancestry-based information. And because the random match probability is calculated, using the frequency of each of these, 16 to 20 alleles in the population, it means that if you match at all the sites, it's like a 1 in 10 to the 15 to 1 in 10 to the 17, chance that it's a random match, which is more than there are people on the planet. So it can be very effective at matching 1-to-1. Assuming that you're in a database, right? But SNPs, on the other hand, are very different marker type. As probably most of you know, most human genetic variation is at a biallelic site, and there are many, many, many sites that differ, of these types of sites that differ with any particular person. But each one of them, any one of them alone, has very low information content. But there are millions of them, and nearly all of them are in LD, linkage disequilibrium, with nearby sites, which gives us an advantage because it means that if I tell you what allele is at a given position, it also contains some information about what alleles should be at neighboring positions. So for this reason, we can use low pass whole genome data and are likely to be able to impute the SNP sites that we need to observe, even if we don't observe them directly. The next difference in these kind of three main technologies or techniques is the database or the searching aspect. So law enforcement at the federal, state, and local levels maintain and control access to the databases that contain these STR profiles. An STR profile is obviously very effective if someone's in the database, but there's a lot of reasons why someone might not be in the database. It's a very select group of convicted offenders or other CODIS eligible profiles that are in these databases. So when you strike out looking for that match, this is where consumer databases can become very useful. Now, obviously, these consumer databases grew very organically. They're either controlled privately by the companies or publicly in the case of GEDmatch, but you can't use STR data. It's not compatible. So you need a completely new data type. You need genotype data. And if there are two ways to get genotype data. There's genotype arrays or there's whole genome sequencing. The last thing I want to kind of compare across these technologies is just the sample limitation. So SDRs are length polymorphisms, and they will fail if the DNA fragments that are in your sample are too short for the amplicons, or for the fragments to amplify, right? Whereas genotype arrays are more sensitive to quality. They fail with less than 10 nanograms. I think they prefer to load 20 nanograms of DNA. That's high quality. So these come from spit kits or cheek swabs. And they're kind of susceptible to failing if there's a lot of contaminating DNA, which is often the case with forensic samples. These are samples that are buried or submerged or just old and colonized by microbes. So I'm kind of being tongue-in-cheek here by saying Astrea has no sample limitations, but I'm going to get into a little bit more about that. So by using paleogenomic wet lab techniques for both extraction and the process of converting those extracts to a DNA sequencing library, as well as our experience working with super low coverage data, we really have this now unique ability to contribute to the field by using a sample type that was actually long thought impossible to work with, and that is rootless hair. So some fun facts about hair. So the DNA quantity in a single hair shaft when it's extracted by us we infer that it's in picograms, but it's actually often completely undetectable by a fluorometer, which is kind of quantifying the mass of DNA. It uses an intercalating dye. So essentially you're looking at the mass of any DNA that's present, human or otherwise. And we often get unquantifiable DNA extracts, but this we actually stopped quantifying them because we can always make a sequencing library and we can always sequence some of the hair regardless of whether or not it quantifies. So that's kind of cool. Another fun fact about DNA in hair shafts is that it's very, very short. And we saw that a little bit when I didn't describe it very much with the Miranda Eve hair, but we're going to look at this here. The most common fragment length are fragments that are well below 100 base pairs. So here is a subset of data that Ed is working on at the academic lab. He's trying to characterize DNA from hair kind of writ large. So these are all hair samples from people living today. The hair that's sitting on your head right now has DNA that is already very, very degraded. So it's got, so we'll look over here, the mean, sort of mean distribution length of, about 40 base pairs or somewhere around 50 base pairs here for mitochondrial DNA. It is also obviously less than 100 base pairs for nuclear DNA as well. This sawtooth pattern, by the way, if you're not familiar with this is a cool feature that DNA, when it's, that sawtooth pattern occurs every 10.4 base pairs, which is the length of DNA wrapped, in a single helical turn. So as DNA degrades and breaks from its structure, you get fragments that are the length of fragment lengths associated with that structure. So head hair, pubic hair, plenty of nuclear DNA, plenty of mitochondrial DNA. We're starting to kind of recognize why this is a myth. But the myth really comes from this observation in the field of forensics. So what I've plotted here are the amplicon lengths of STRs and mini STRs, which were developed to try to kind of accommodate this problem where some forensic samples have very short DNA. But even with mini STRs, almost all of the markers that one needs to assay for an STR profile in order to upload it to CODIS or one of these law enforcement databases, almost all of those markers are too long to amplify the DNA that is in hair. So that is where this longstanding myth came from. It is the reason why a lot of forensic scientists will say or would have said there is no DNA in hair. There is no nuclear DNA. Another fun fact is that DNA preserves very well in hair over time. It's an insoluble kind of substrate. It doesn't uptake contamination as much as, say, bone or teeth. And so what we're looking at here is hair that was sampled at the, off of my head. And also I had hair that was 23 years old that I had collected as a kid. And so you start to see that the most common fragment lengths begin to be shorter and shorter. And then in red is the Miranda Eve hair, so 140-year-old hair. So it just gets shorter and shorter, but it doesn't go away. It just gets kind of harder to deal with computationally. It's still there. Another kind of fun experiment is that we got some hair from, or some fur, some hair from extinct mammoth and an extinct rhino that were over 40,000 years old. And it has a really similar DNA length profile as the hair that's on your head right now. So this is a sample type that we can work with and is exciting for kind of cold cases because it's often a sample type that was collected and then not really handled. So if a hair shaft didn't have a root, it was usually set aside and not tested. The root, sometimes contains some actual cellular material. So this is just some fragment length plots. This is in log scale, so it looks like the shape of the distribution is a little bit different, but it's really short. You can be rest assured that it's going to be mostly under 50 base pairs. Okay, so the process overview that we do at Astrea is we take a bone or a tooth or a hair or whatever the sample is. We do sort of an ancient DNA-like extraction method. We sequence it, we call the genotype, and then we pass off the data back to law enforcement, and they enlist the help of their genealogists in order to try to build out those trees the similar way that I showed with Miranda Eve. So kind of our role stops after producing a genotype file. We do a QC pass-fail before genotyping because genotyping still requires a lot of data, and I'll kind of explain that. So I'm going to go through just briefly. We kind of have as I mentioned, a paleogenomic approach. We've worked with a lot of samples that are hundreds, thousands of years old. So kind of take a boutique kind of high touch approach. We want to understand how the sample's been, how it was found, how it's been stored, what kinds of elements we have been dealing with, humic acid, what kind of inhibition, things like that. And we can kind of cater our approach to getting the best DNA and the shortest DNA possible out of the sample at the extraction stage. But then there's the method of getting that DNA into a sequenceable form. And for that, we use this seriously library prep method. This is a single-stranded library prep method. This is good for degraded DNA because if you have single strands of DNA or nicked double strands of DNA that are floating around in your sample, if you use a traditional double-stranded DNA library prep, which is the most common kind of library prep, you will lose those single strands. And if your sample is heavily degraded, you might be throwing away a lot of very useful information. So you kind of gain with using the Astrea workflow, you gain single strands, you gain access to double-stranded DNA that might have been nicked. You also still get the duplex DNA. It's not like it's going anywhere. You just are observing both the top strand and the bottom strand separately. So this is how we sort of do this in the wet lab. And then there's the QC analysis, where we kind of estimate how many unique molecules we have in the hair sample or in the forensic sample in order to kind of get an idea of, do we have enough data to call genotypes? So if we were to sequence a lot of data, if we were to be able to obtain a lot of sequence data, like 30-fold coverage of the genome, which is totally easy with fresh, high-quality samples, it's like really common, say, in clinical genomics, it would be trivial to call genotypes because every place that you want to call a target SNP you've seen it on average like 30 times. So you can make pretty easy inference about what the genotype, what both mom and dad's alleles were. But in forensic samples, we almost never achieve that high of coverage. What we typically see is something about twofold coverage. So what makes genotyping possible for us is that we actually only need to impute the positions that are on the direct-to-consumer arrays, because those are what's in the database that the genealogists need to compare to find relatives. Now, if we think about this in sort of a population genetic tree framework, which is a kind of a good framework to think about these kinds of things, the most common variation is paradoxically rare variation. It's unique to you and your close family. It's very low down on the tree. It means that with every new variant born, it's born at very, very low frequency. The only way that a mutation reaches high frequency is through time, is through being passed through in generations. And the lucky fact is that the SNPs that are chosen for these arrays have high alternate allele frequencies. They're way up in the trees. They're it's sites where it's common to either have the common or the rare thing. So that feature is something we can take advantage of. It means that we can infer the genotype we can use these linked sites that are low down in the tree to infer the mutation that is high up in the tree, which is the ones that are on these arrays. So I think for every target site that we're interested in calling a genotype for, there's about 40 or 50 linked sites nearby that contain information about what that allele could be. So I'm not sure how much detail we want to get into how our imputation software works, but what we're doing is understanding haplotypes, common haplotypes that segregate in the population for every single target site. And we learn that information from things like 1,000 Genomes Project, and then we can take the data that we do observe and ask, well, what is the likely genotype at this position? So how well does it work? How accurate is the imputation software, and what can we do with them? So here we're showing this with a test individual. So this is our accuracy at twofold coverage when we compare genotype calls that are made using this imputation or genotype refinement method, compared with genotype data that is generated with the genotype array. And what you'll see down at the bottom, so shown in blue, are the 5 closest relatives that match in the GEDmatch database for this individual. When the array data is, when the data come from a genotype array, so that's blue, versus in yellow, where the genotype data comes from whole genome shotgun and imputation. And what we can see is that the top 5 closest relatives and the degree of relatedness, so the total centimorgans of matching DNA is similar regardless of the way that the individual was genotyped. So Ed has tuned our software currently so that 9 out of 10 times we make a call that's right. 9% of the times we don't make a call at all. And at that tuning, we get about 1% incorrect calls. So we can tune this, of course, to make more correct calls at the expense of making more incorrect calls. But this particular tuning works well for what we're being asked to do, which is, find relatives in a data in these open source databases. Okay, so here is an example case. This is a Jane Doe from the 1970s that was referred to as Precious Jane Doe. James Scharf. A lot of the cases we get, being cold cases, are usually motivated by one detective who sort of sat with the case for years and years, and they're paying attention to the technology developments, and they're a force to be reckoned with. They're driven to get the funding and apply any techniques that are possible to these old samples. Jim Scharf has done that with Precious Jane Doe. So this is a kind of rare case where the murderer was known, but the victim was never ID'd because he didn't know who she was. She was a hitchhiker that he picked up. And so this is kind of an interesting, the crime was solved. The murder was identified. He confessed, was convicted. And all this time, no one knew who the actual victim was. So they had done some early attempts to generate leads, using skeletal remains to estimate age at death and doing some sketches. And before the Golden State Killer case actually broke the news, this case was also the subject of some of the first attempts at doing forensic genetic genealogy. So there's this article published in 2017 describing these early efforts to extract DNA from the girl's remains in an effort to provide a genetic profile that could be used for searching for relatives in the database. This sounds really familiar, right? Unfortunately, all of those efforts failed because the remains were in such poor condition that they couldn't get enough clean DNA extracted or analyzed to make a DNA profile for upload. And some of the best labs in the world worked on this, mostly with skeletal elements, and no one was able to get enough DNA for a genotype or SNP profile. So then Jim Scharf, through an introduction via Barbara Ray Venter, he had contacted Ed's lab, and this eventually came over into Astrea's lab, but connected and said, I have some hair from this individual. Can you try it out? So I'm going to show you some of the results of that. What we're looking at here is just the read composition. So this is saying of all the reads here in purple, of all the reads that were sequenced, how many of them were short enough to be merged. This is paradigm sequencing. How many reads were discarded either because they were adapter dimers or they were just too short to be reliably mapped to the human genome. And then over here, we're saying, well, of those merged reads, how many of those reads could be reliably mapped to humans? So the yellow is here. So, 90% of the merged reads are human. So this is already a really good sign. We can look at this and know that the DNA reads look like hair. They're very, very short. So you're looking at the purple line here. This is merged reads that are also mapped to the human genome. We can also tell that, of course, she's a woman or a female, biological or chromosomal sex as female. This is just kind of taking the proportions of, females have 2 X's, and chromosome 10 is a similar size to chromosome X. So you would expect to see, in a male, you would expect to see half the amount of DNA mapping to the X as you do to the autosome, and a little bit more in Y. So when you see double the amount of X or equal amount to the autosome, which has 2 copies, then you can tell that it's a female. Then library complexity. This is important because what we're doing here is trying to estimate at first how many of the DNA fragments that are human, how many are unique, how many are duplicates. Because what we need to be able to do is get about 2-fold genome coverage. And so we use this complexity estimate to try to accomplish this. This is a pretty highly complex library. This is saying that if you sequence 300 million reads, you are likely to see 200 million of them be completely unique molecules. So 300 million DNA sequence reads were used to call 1.4 million SNPs, overlapping all of the different direct-to-consumer panels with a genotype file that was ready to upload. And so, so I was going to show, I think I hid this. I was going to show what a match looks like on GEDmatch. What you're observing when you're matching somebody. And what I was showing was an example where the top match to the hair sample is about 300 centimorgans. And so then you can look at a chart like this is what the genetic genealogists do, and they say, okay, with about 300 centimorgans of shared ancestry, what kind of a relationship could it be? And then they start going and building out the genetic or the family trees based on this kind of information. That's not part of what we do, so it's a little bit limited what I can say about how difficult it is. I'm sure it's very difficult and time consuming, but happy to report that the DNA was able to provide a file that matched to a close enough relative that they were able to identify Lisa Roberts. Lisa Roberts was a very difficult case genealogically because her birth family had died in an accident shortly after her death. So not only was she adopted, but she had been a runaway, and she had been missing, reported missing, but then the initial report was kind of clawed back for unknown reasons. So she kind of fell through the cracks, and they were able to name her again as Lisa Roberts, and she had several, she had half siblings that remembered her and were obviously very happy to know what happened to her. So we have several success stories now. We don't always know when a profile we provide is successful. Sometimes I just Google Astrea Forensics and see what comes up and see what news reports I can find. So sometimes we have relationships with the genealogists that let us know. Sometimes the detectives are sending us Christmas cards. It depends on the kind of relationship we have with the agencies. But we've now, since 2019, we've taken on, by now it's probably 80 something cases where we're involved in some way or another, whether we're just doing extraction, whether we're doing the whole profiling. So the take home message is direct sequencing provides information about a sample that is unattainable from other types of technologies. And rootless hair is a particularly well suited sample for this. And we hope that all kinds of agencies learn about us. And so I think with that, I have a lot more slides, but I think best to probably go to questions, and maybe some of the slides that I have will be useful in discussing that. So I'd like to thank you guys for your attention. Thank, of course, the team that kind of is in the lab making this possible, and the founders and the genealogists and the detectives, specifically Jim for this case.
That was fascinating. Bits of it over my head, but just all around fascinating. Yeah, if you have questions, please enter them in the Q&A section. And looks like we have a couple of them already. Do you want to read them?
So does CODIS have information about unsolved crime samples? I don't know. Because I have being a database that is tightly controlled by law enforcement, I'm not sure that that's the source for information about unsolved crime samples, but there's a database called NAMUS, NAMUS, and that is a list of, if the agency participates in uploading to NAMUS, that's a list of every unsolved or unidentified remains that's in the United States. There's thousands and thousands of unidentified remains. And not all agencies choose to upload that information. So there's probably a lot more than that. How does bleaching or coloring hair in the DNA change the DNA in the hair? This is something we're actively trying to understand. So one of the interesting things about hair is that within a strand and between strands on the same head of hair, there's variability in the amount of DNA that's preserved. It's just the variability of like how many cells kind of nucleated, like just traveling down the length of the shaft. So we've done tests of hairs that had been color treated and those that have not. And sometimes the results are exactly the same between the two hairs and sometimes they're not. And it's likely more about human variation than it is about the treatment. However, if hair is sitting out in the desert, for example, for a long time and under constant UV irradiation, then it is likely going to be really, really damaged to the point where most of the DNA won't be usable. And I probably have a slide that would show that, but you mentioned centimorgan distance, but not number of shared segments, which 23andMe reports for relative comments. So centimorgan distance is, so GEDmatch is kind of the only database I'm familiar with because it's the only one I have access to see. And they report total centimorgan matches, which is just a count of the accumulated smaller segments. So you can have several large fragments that equal the total shared centimorgan. And what that means to me as somebody who's sort of trying to evaluate this in a whole genome data, is that if the individual shared segment is large and contains an appropriate number of SNPs, it's likely real. Like I guess there's parts of the genomes that we are more likely, any two people are more likely to match to, because it's just a part of the genome that we share in common among a lot of humans. So yeah, the number of shared segments just adds up to the total centimorgans, and then the total centimorgan is how those relationship charts work. I hope that answers that question. Do you treat extracted DNA with repair enzymes? No, we don't. We could, but we have found that we don't need to, because this single-stranded library prep that we use can convert DNA in its sort of native form, regardless of whether it has damage, abasic sites, depurination, deamination kind of in or around its fragment ends. We can still convert those to good sequencing libraries, and some of the algorithms we have do take into consideration certain damage patterns when sort of doing alignment and doing sort of consensus sequencing kinds of things. So we don't. Does the recent work, so I won't be able to answer this because I am totally unfamiliar with it, indicating the age of the sample effective approach. Sorry, it doesn't affect our approach because we don't know about it. But I'm going to write that down so I can look it up. Different ancestry groups have different centimorgan relationship mappings, like Ashkenazi Jewish ancestry has higher similarity between fourth cousins than other European populations. Yeah, okay. And that is kind of one comment that makes me think, I've heard from, this is kind of totally a segue, but the genealogists that are being used in to do some of this relative matching with these databases, they're kind of coming out of the woodwork, right? Genealogy, genetic genealogy, genealogy at whole, in general, is one of, if not the biggest, most common hobby in the world. So there's a lot of genealogists who are getting into this kind of industry, and there are certain states that are now beginning to regulate the kinds of qualifications that a genetic genealogist has to have, which is going to be very confusing because there are no qualifications right now that are available. So it's a good idea, but it's going to be hard to implement. And the reason I'm bringing this up is it's good to have a good genealogist, because they'll know, they'll understand these kinds of things about different ancestry groups having different kinds of relationships and how that affects the relationship chart with the amount of centimorgans. Those centimorgans are a range anyway. So I think some of that information is oh, sorry.
So yeah, we should probably read the question. Next time I'll post the questions in the chat just so they're written down. I noticed that very interesting chart you had, by the way. No more open questions, so I'm going to just ask one last one. Where it showed the sort of the ancestral relationships between people, and then the fraction of the genome that was shared. But surely there's a little bit of redundancy there. Like if you share a certain fraction of your genome, then you could be either this kind of cousin or that kind of cousin or whatever. How do you distinguish between those relationships?
So that is all, I mean, that's why genealogy is so hard. If you have a match that's, like 3,700 or, 3,000 centimorgans, it could be a parent or a child, right? It could be, things that 25% could be grandparent or or sibling.
But usually which one of it's going to be, right?
Yeah, and then once you get further out, then I mean, it'd probably be good to just share that screen just briefly again, just for
because family trees, of course, as Kevin just mentioned with these Ashkenazi groups, with a lot of sort of in the word inbreeding, right? Maybe not the right word, but family tree crossing, right? Right. Like
so this is an example of what you mean. If you especially if you don't know what generation you're in, if you have someone matching at 300 centimorgans to your sample, that could mean any of these relationships that I've circled. So then the genealogists have a lot of work to do to build out each of these trees with each of these hypotheses in mind. So now these are all hypotheses that are to be tested until they're ruled out. And that's why the genealogy part of this is so hard.
Great. One last question. I know we're at time, and I thank you very much for talking today. Where do you see your business scaling? Like, I guess you just mentioned there's just more and more and more of these cases waiting to be solved. How are you going to scale up this really sort of intensive, basically, service that you're providing?
Yeah, that's a great question. I mean, right now, the sequencing in general is so much more expensive than the direct-to-consumer assays. That's something we didn't talk about. As a limitation of what we do, you can do a $100 direct-to-consumer test, or you can do a $10,000 whole genome test, right? Totally different scale. Given that there is growing appetite for being able to kind of spend funds to do that, our next steps are, can we use different kinds of approaches to make it cheaper so that we can scale it? And then it's all about automation. So there are some parts of the process that we do, like I mentioned, are kind of boutique or high touch. And I think for the extraction part of the process, that'll kind of have to be true for a long time. But there are other parts of the process that are very easily automatable, and that's probably where we would head. The other interesting thing I want to mention is that we only perform this work for law enforcement. We have a very constricted terms and conditions of service, but it would be really cool to be able to offer this direct to consumer someday. And there's a lot of people who I have my grandmother's hairbrush, and I was able to get her understand her place in my family tree. That would be a cool service to be able to provide, and one we're definitely thinking about how we can get costs down to the level where we can approach consumers.
Great. Thank you, everybody, for watching today, and thanks very much, Kelly. Fascinating. And if I hope people can get in touch if they have specific questions. I'll forward any inquiries your way. Leighton has one last point, but it's really you'll read it, I guess.
Yeah, probably. And I can address Kevin Carbos's too, but I can do that. Yeah, I can write him an email.
Great. I think we should end it here. Thanks, everybody, and especially to you, Kelly.
Okay, thanks.
Bye now.